On this week's episode of the Galactosemia podcast, we talk about all things test results, the different numbers you might see on your test results when you test for galactosemia, including gene variants, how Galactitol and GAL1P are measured. We talk about false positives and newborn screenings, and a slew of other things. Our guests today are experts in this field. We have a clinical geneticist from Emory and a lab director from the Mayo Clinic, so really no better people to talk about this with. It really was one of my favorite episodes. I felt like I learned so much. I actually have a much better understanding of the tests that Elliot undergoes and what those results mean. I had to listen back to the episode a couple of times to fully grasp everything, so don't be ashamed if you have to do that too. That's the beauty of a recorded podcast. You can listen as many times as you need to. It's also worth noting that our guests shared some really great visual slides during the episode, so I highly encourage watching this episode on YouTube if you are able to, especially if you're a visual learner. Seeing some of the things that were talked about can make it so much easier to understand. If you enjoy these episodes, consider subscribing at thegalactosemiapodcast.com. It really helps us keep this going, and we put all the episodes up there ad-free and at least a day early. Okay, let's jump right in. Please enjoy this episode all about reading galactosemia test results with Dr. Tricia Hall and Dr. Michael Gambello. All right, I have two guests with me on the show today, and I'm really excited to talk with them about something that, I know a lot of us as parents tend to wrestle with, and that's understanding the test results and genetic reports that come along with the galactosemia diagnosis. So my guests today are Dr. Michael Gambello, a clinical geneticist at Emory University, and Dr. Tricia Hall, a biochemical genetics lab director at the Mayo Clinic. Dr. Hall works directly with the galactosemia lab results, literally signing out what shows up on our kids' patient test results. And Dr. Gambello has spent over 25 years working with families of children with metabolic conditions, helping them understand what all of this actually means. Michael and Tricia, thank you so much for joining me today.
Happy to be here.
Happy to be here. Yes, thanks so much, Ryan.
Of course. All right. So before we jump in, let me give you both a little bit of context on my story, because I think it kind of helps understand where I'm coming from and my lack of knowledge, hopefully. My son, Elliot, was diagnosed with classic galactosemia in August of 2024. And like most parents, I had never heard of it before. It was the first time I'd ever heard that word. And I have since thrown myself into learning everything I can via this podcast mostly. And, one of the things that became a bit of a rabbit hole for me were test results, especially as a first-time parent. Honestly, for me, I find it hard to make sure that I'm going to the doctor enough myself, getting tested for things at 37 years of age. Like, I should probably be going more often. And now I'm trying to understand gene variants and GAL1P levels and Galactitol levels, and it can all be very overwhelming. And so I want to get your help on understanding some of that stuff. But before we jump into it, I'd love for you both to give us a quick overview of what you do and how you interact with galactosemia patients and their data. And Tricia, if we could start with you, what does it mean to be a lab director, and how do these results come across your desk?
Sure. So as a lab director, we are responsible for oversight of the operations of our lab. So the scientific, the technical, and the clinical oversight of test results when they come in. For a lot of the genetic test results in a biochemical genetics laboratory, they're reviewed by a laboratory director before they are released to patients and providers. So we basically take a look at the results that our technologists, they will receive samples, process the samples, make sure that the results meet our quality metrics, and then they'll come to me for review. So I'll generally get, we usually do testing in batches, so we'll have however many results have shown up on that day and are processed. We get our quality metrics that we know the assay is working as we intend it to. Patient demographics, we can look up previous testing to make sure anything isn't considerably out of range or discrepant with what things have looked like in the past. And then also just to ensure that the results meet quality metrics, they clinically make sense. And then we're going to sign them in our laboratory information system, and then they will get released usually to the ordering provider and their laboratory information system, and then they generally will filter through to like patient portals and where you guys can directly see results.
Got it. And are when you're seeing the results of the tests that are being run, are you making assumptions off of those tests?
Not so much assumptions as interpretations. So
Okay.
typically when we're doing enzyme results that are diagnostic, we will be able to have kind of a few scenarios where we have the group of patients who are, clearly newborn screening follow-up. So they're all a week to 10 days old and they're getting, testing for the first time, and they usually have some degree of reduced enzyme activity. So then we're kind of putting that on the scale of classic galactosemia to carriers and other potential genotypes that you can detect by newborn screening, and looking at those and interpreting them and making sure, and then also correlating those, because we'll often receive galactose 1-phosphate or Gal1P levels at the same time, and then kind of seeing who is where they should be, and does the enzyme correlate with their Gal1P, or is it very strangely off, and we should probably dig into that a little bit more. Are we looking for a different form of galactosemia? And kind of just correlating, and less assumptions and more just interpretations, and putting everything in the right context and making sure it all makes sense, so that we're not releasing anything that's not as accurate as it can be.
Got it. That makes sense. And Michael, to move to you, you're a clinical geneticist at Emory. What's your day-to-day like, and how do you use those lab results in your work with families?
Sure. So on my clinic days is when I see those patients. I'm sort of an administrator. I'm sort of an email doctor these days, but I do still see patients. And for the kids who have positive newborn screens, as Tricia was saying, it's imperative for us to do follow-up testing, and that usually involves DNA testing as well as enzyme level and Gal1P. Galactitol is interesting, and I was talking to one of my colleagues, Judy Friedrich-Kyle, who does a lot of basic science work on galactosemia, and Tricia could probably chime in here, but I don't think people are using galactitol as much as they're using Gal1P, I think,
I would agree with that.
in labs around the country. Um, I don't fully understand why, because
That was going to be
galactitol
my
just
next
requires
question.
you to pee in
Yeah.
a cup, whereas Gal1P, it's actually Gal1Ps are a little bit more involved, even in the processing. So at any rate, we would send those labs, and then when the results come back, we would interpret them, looking at the interpretation of the lab, and then explain the results to the family if it in fact indicates the child has classic galactosemia, or sometimes we pick up carriers. We can talk about that later, and sometimes it's other forms of galactosemia, as Tricia was talking about.
And is it your experience that those decisions on whether to use galactitol as a marker or to test for Gal1P or both, does that recommendation typically come from the genetic doctor? And should the parent have a say in that?
That's a good question. I think it's typically what the clinic is used to doing, right?
Okay.
Like, at Emory we do Gal1P.
Mm-hmm.
At Cincinnati Children's we do Galactitol, right? I mean,
Right.
and we could get into um, what those values actually help
Mm-hmm.
with. I think they simply help with making a diagnosis and looking at dietary compliance. There's not a lot of correlation between the levels of Gal1P or galactitol and how well a child does.
That's really interesting, because I think, especially as a new parent, that is something you kind of assume that, like, oh, if my son or daughter
Well,
has higher levels, they're going to be more affected.
right. And so, studies have been done, and Judy just did a study that really doesn't show a significant correlation with blood Gal1P. Now, who knows what the brain Gal1P is, right? There may be correlation with brain Gal1P, but obviously we can't measure that.
And that's something I want to talk to Dr. Judy about, a frequent friend of the podcast, has been on a couple of times, and I'm getting ready to talk to her again in a few weeks, and we're going to talk about some of the genetic breakthroughs that have happened with CRISPR and, the story that came out in the New York Times. But one of the things she pointed out was there are different areas of, I don't even really understand it, this is why I'm going to talk to her, but like you said, like brain Gal1P versus this Gal1P versus there's a lot of different areas that it lives. So that's really interesting. Moving to I actually wanting to understand the test results and stuff. The best way for me to typically understand is to have a hands-on experience. And so what I've got for us are some actual test results from galactosemia patients that I would love to dig into with you guys. But first, before we do that, I want to start, if that's okay, with a little genetics 101 for parents who barely passed biology. And if you'll allow me to, I may interrupt you here and there from time to time for explanation, because asking you to break down these things on a more elementary level is probably key, because whenever I say for parents who barely passed biology, I'm really referring to myself and my very minute level of understanding of how genetics works.
Fair.
So I think if we could start with gene variants, that would be a really good idea. And I would like to start with, we hear Q188R, L195P, C584, more than C, and I feel totally lost. I imagine there's an entry level to even before you get to variants, you kind of have to probably understand genetics. So, Michael, could you kind of help us out there?
Yeah, so let me share my screen.
Great. And if you're listening on the podcast, Michael put together a little presentation for us. He's going to share his screen. I encourage you to go over to YouTube. There are some things he's going to share with us there that you can visually see.
So I just want to make sure everybody understands what galactosemia is. So
okay.
For the most part, let's say we're talking about classic galactosemia, the fairly severe disorder.
Sure.
And it is a defect in an enzyme or a protein in the body. Okay. And this protein converts one chemical to another chemical. So you'll see here, galactose 1-phosphate is converted to glucose 1-phosphate by this enzyme that we call GALT. We're not going to tell you what the full name of GALT is because it is too overwhelming. So we just like to call it GALT. So it converts the galactose 1-phosphate to the glucose 1-phosphate. Children who have galactosemia have almost zero GALT in their body. And so you see the X there. So they can't get rid of their Gal1P efficiently. So their galactose 1-phosphate goes up and they don't get as much synthesis as glucose 1-phosphate.
Would you say the GALT enzyme is not there at all, or would it be more accurate to say it's there, but it's not functioning correctly?
Well, it depends on the mutation. So
okay.
That's sort of advanced genetics. So
we're going to get there, hopefully.
So you can have enzyme there, but it doesn't work. And you can also have no enzyme there. And Tricia could chime in here. When she does her testing, she's looking to see if the enzyme works.
Correct, yes.
Okay. Are you looking to see if there's an enzyme there?
For practical purposes, with a test that we do, the enzyme not working and the enzyme not being there looks the same.
Okay. Got it.
And I think it would, Michael can correct me if I'm wrong, that has the same clinical consequences.
Yes. Okay, so that's what galactosemia is. So now it turns out that all the proteins in our body are coded for by genes.
All right,
so there is a GALT gene that we have in every single cell of our body. In fact, we have two copies. We have two copies of most genes. We get a copy of GALT gene from our mother and a copy from our father.
So our body is made up of cells, and these cells, every single cell contains a copy of the GALT gene, 2 copies of the GALT gene.
That's correct.
Okay. So this process of moving from galactose 1-phosphate to glucose 1-P is happening in every cell. Is that accurate to say?
Well, that's probably not true.
Okay.
They all have the gene, but they don't all have the functional enzyme expressed. So that's more liver, red blood cells, where those things are happening. Not necessarily things like bone or anything like that, where this enzyme reaction is not happening there. It's happening more in the kind of more active cells of the body.
Got it. So there's specific places in the body where the cells will need to activate this GALT enzyme to convert galactose 1-phosphate.
Yes.
Okay. Got it.
Okay, so let's go to a figure that shows what genes look like.
Okay.
I think we're going to jump right here. Okay. So
very confusing. What I'm looking at is just a bunch of letters
that's
and
right.
shapes.
So I'm going to make it all clear to you.
Okay.
In 5 minutes, you will see the light.
Okay.
So a gene is a string of letters. There's only 4 letters that are used in biology: A, T, G, and C. They stand for specific chemicals. And there is a string of A's, T's, G's, and C's that code for a specific protein. And the genetic code is read in 3 letters at a time. So, for example, if you look at the example here, this gene, this is a small gene, but it has ATG, CAA, CCT, CCT, and CGG. Okay, so that's a small gene. And what I'm showing you underneath is the protein that gene is coding for. So in the cells know that an ATG should give you this M. So these are building blocks of proteins. They're called amino acids. Okay, so you have a small m coded for by ATG. A small q is coded for by CAA. A p is coded for by CCT. Same thing here. And then the last amino acid is this R.
So do these in the cells, does every ATG equal an M in a protein?
Yes.
Okay. And then are there some genes, you said this is a small gene, it's made up of, it looks like 3, 6, 9, 12, 15 letters. Are there some that are 18, 21, 24 letters?
1,000.
1,000. Okay.
Yeah.
How is it accurate to ask how many genes there are? Is that a thing?
Well, yeah. So we actually have a reasonably good answer. At least the last time I heard, we have about, 20,000.
Okay.
Tricia, what's your last count?
I thought I would have said between 18 and 20, but that we don't know what they all do.
We don't know what they all do.
No.
But some of them we do know what they do, and in this particular instance, this gene, ATG, CAA, CCT, CCT, CGG...
This gene that I'm showing you is an example gene.
Okay.
Okay? This is not the GALT gene. This is just an example of a gene.
Okay. And so this gene, you said it's coded to does it make the protein? So
basically, the genes contain all of the information to make proteins. And it's the cell that has a complicated mechanism of reading that code and translating it into protein.
Got it. Okay, so the cell reads ATG and it goes, the first part of this protein is M.
Yes.
And then it reads CAA and it goes, the second part of this protein is Q. And our cells are doing those essentially with like somewhere between 18 and 20,000 genes?
Pretty well, yes, they're doing a lot of that.
Okay, wow. Okay. And so then that protein that you get is MQPPR.
Right. And, okay, again, this is a hypothetical protein. But in real life, we would have the DNA sequence of GALT, and then under it, we would have the amino acids that are strung together to make the GALT enzyme.
Okay. Is the reason we didn't use that in this example because like
because it's too big. With the slide,
huge.
wouldn't we would have, they would go on and on and on.
I just looked it up. The GALT gene has 12,000 bases. So that first line, the ATGs, that's 12,000 in GALT.
So ATG has 12,000 characters in it?
No.
The gene would have 12,000 characters if we put the GALT example up there.
Right. So in other words, it would go ATG, C, and all these letters would keep going on and on and on, filling up about like 12 slides.
Got it. Okay. But that, you said, essentially our cells read that GALT enzyme that is made up of those 12,000 characters, and it what does it do then whenever it sees that GALT enzyme? Does it make a like in our body, we don't have classic galactosemia. So in my body, it reads that and it turns it into a protein.
And then the protein finds some Gal1P and converts it to glucose 1-phosphate.
Okay. I'm starting to I have seen the light, I think, as you called it. That makes a lot of sense. So when we're talking, is it now the next step to talk about a variant?
Yeah, so now we can talk about, if you want, Q188R. I actually have a slide with one of the reports that you wanted to look at.
Uh-huh.
So you see up here, it says, this is the gene they're looking at is GALT, and it says C584T to C. And then it says PLEU195 in parenthesis P.L195P close parenthesis.
Yeah, and so just to break it down for anyone listening, what we're looking at is an actual result, a test result that I got in touch with a few patients and put together some redacted test results. So this is an actual test result from someone who has classic galactosemia, where it's showing the gene, the variant, the zygosity, and the classification.
Right. So you'll see, then it has zygosity. We can talk about that in a minute. And classification, pathogenic means it's disease. It's a disease-causing change. Okay. So this is all genetics nomenclature. It's sort of the way we understand what's going on. So let's go back to our example. Okay, so this is looking at our example a little differently. So it turns out that we number genes where the first letter is 1, the second letter is 2, the third is 3, all the way until the end of the gene.
Okay.
So
in a GALT gene, there's essentially about 12,000.
Correct.
It numbers up to 12,000.
That's correct. So now this was the little Dr. G's example gene.
Yep.
And this is the protein that gene makes. And we can also number the building blocks of the protein. Number the amino acids, or the building blocks of the protein are also numbered 1, 2, 3, 4, and 5.
Yep.
Okay, so look beneath here. If we change, so notice the fifth letter is an A.
Right.
But notice down here, there's a red G in its place.
Right.
Okay, that's a mutation. That's a change.
Okay.
And what that change does is, instead of the cell recognizing this code as a Q, it puts an R there.
Oh,
okay.
Because CGG is coded to
That's
R.
right. So because we've changed the code.
Right.
And there's a way we write that.
Okay.
Okay, so if you look next to the gene here, I have C.5 A to G.
Okay.
Because that means in the fifth letter position, the A was changed to a G.
And what does the C mean?
Okay, so the C means it's the coding part of the gene.
Okay. Okay. The coding part of the gene. Cool. That's funny that's like, we're going through all this stuff and then we're like, we're going to do C for code. Like, okay, cool.
Okay. So that's the DNA change. That's the gene change.
Okay.
And then this is the consequence of that change in the protein. That's the P.
Is protein.
The Q at position 2 is now an R.
Okay. We didn't want to use a little less than or more greater than symbol to keep it consistent between
Yeah, we don't
gene
do
and
that.
protein. Okay.
I don't know why.
The first person didn't do that, and
Yeah.
everybody followed suit.
We don't need consistency for the sake of non-confusion. Okay, so yeah, can we go back to your...
So let's
test
go
result?
back here. So now it should make sense. So basically,
Let me try it. Can I try it?
yes, you try.
So in the coding part of this variant, the coding part of this gene, the variant is at, I'm trying to blow up my screen, 584th position. The T has become a C. I'm still a little bit confused at the PLEU part.
Where's? Oh yeah. Well, we'll
underneath.
explain that.
Okay.
I could
But
let
that's-
Tricia explain that one.
Okay. So far that's it though. That's what we're saying is like this galactosemia variant is that in the GALT gene, we've figured out that the 584th position, it should be a T, and instead this person has a C.
Perfect.
Wow. Okay. And that is meaning, and that the consequence of that is that the protein is not functioning correctly.
Well, that's correct. But also, if you look underneath, you'll see in the parenthesis it has PL195 to P. So I was using single letter codes, but there's also the 3-letter code. So LEU stands for leucine, but also L by itself is leucine. I have to say they don't make these easy for lay folks to understand.
Yeah, I've completely gotten lost already.
And
I still remember the
third
first part.
line, we're not even going there.
Okay.
Because I don't think we need to.
Okay. So the first one is your gene variant, how it is coded, what the T becomes a C. Is there, you had a different thing, which was the consequence of P. Is that what I'm looking at on the second line here?
Yep.
Yes. Yeah.
Okay. So P, it would be, in your example, L195 Pro. Can you refresh my memory? I've already forgotten what that means.
So pro is it's proline, and if you look in the parentheses following that, that's the nomenclature Michael had on his slide, which is the kind of single letter terminology. So P dot L to at 195 to P. So that's leucine at 195 to proline.
Right. So your example was PQ2R, which was P, the Q in the second position. I guess where it trips me up is, yeah, like in the first one, it's number, letter, and this one we're going letter, number, letter with no greater than sign, but we're essentially saying the same thing. We're
Yeah,
saying,
it's the same thing. Like
okay.
Like Tricia said, it's probably because that's the way the first people who did it.
Right. So we're saying that the code of 584 instead of a C or instead of a T, they have a C. The result of that is that the L in position 195 becomes a P. Leucine becomes protosine.
That's right.
Yep.
Proline.
Nailed
Now, you nailed it. But so the important thing is, what does that mean? Like, does that change cause disease or not? Right. So it's the laboratories that have the data that can eventually say the classification. It says pathogenic. So in other words, this change is in addition to a change on the other gene, right? Because there's two copies.
Okay.
Right, can cause galactosemia.
So are there changes, like could I feasibly have changes in my body that are variants that do not result in disease?
We all do.
We all do.
Exactly.
In all sorts of genes, we all have variation that is just not doing anything of any significance.
And it just so happens that if you have gene variants that match up in a specific way, which we're saying that these laboratories have all of the data on, we can tell like this is going to result in galactosemia.
Usually. I mean, again, our data sets are good, but they're not perfect. And so we could get into that maybe later.
Okay. Tricia, you deal with these on a daily basis. Is there anything worth adding in from your point of view on these and how you guys look at them?
I think from our perspective, we usually get these from a different laboratory. I'm not a molecular geneticist, which they would do the sequencing for this type of testing in their laboratory. We usually like to see how this correlates with our test results, and that kind of comes into when we talk about classifications. Galactosemia is actually a very nice example where we can kind of see the classifications correlate with enzyme directly. So you can get approximately a 50% reduction from one classic allele, so one of those classic variants. And we can see that on our testing. So we can kind of measure carriers and tell whether they have one or two. So we usually correlate a lot with the results of genetic testing to make sure, one, that everything matches, and then two, when Michael was talking about how some of the times we don't know what variants do, oftentimes we can have evidence of what they do in the enzyme test, because the enzyme will either be normal or reduced or absent, and that can help with understanding the results of these tests. So everything kind of works together for a lot of genetic testing.
Got it. That makes sense. Okay, so if we use an example which we hear a lot in the galactosemia community, which is Q188R, I'm going to take a stab at it. It's I guess technically it would be P.Q188R because it's letter.
Nailed it.
Yeah.
Okay, so that means that the Q, I imagine, stands for like Qulene or something.
Oh, no,
No, it actually stands for glutamine.
It's glutamine. It makes no sense whatsoever.
No sense. Okay, glutamine at position 188 is turned into R, which is
Arginine.
Okay, and that typically, in combination with another variant, creates a very significant case of classic galactosemia, which we have seen have pretty bad side effects in a lot of cases.
Right.
Q188R.
So I have patients who have two copies of Q188R, one from mom and one from dad.
Mm-hmm. And do you know if we see variants? Because we said earlier that typically there's no correlation, or there doesn't seem to be a lot of correlation between levels of Galactitol and Gal1P levels and severity of speech and sound disorders or delays, et cetera, et cetera. Do we know if different genes, so having, for example, two copies of Q188R, if that results in more significant delays or in any specific ways?
Generally, it's a little bit tricky to see from the biochemical perspective whether they correlate, because there's also a lot of variables once you start, adjusting diet, and everybody does that in a slightly different way, and everybody reacts to it in a slightly different way. So correlations are very tricky to understand, to see from the lab perspective. Typically what we'll see is kind of more of a linear perspective for single individuals, and following that through time is a little bit more informative than necessarily trying to draw conclusions from the lab results on a kind of a broad basis, just because there's a lot of other factors that go into all of the outcomes and all of the lab results as well.
Okay, got it. And Michael, I know you said you don't want to get into this third line, but could you give us an overview on like what even the third line is?
The third line is
you could show your chromosomes and then you could just map it to there.
Yeah, so the third line, so basically it shows you what, let me go back. So it turns out we were talking about in every cell of the body, there's 46 chromosomes, and the chromosomes are numbered.
What are chromosomes? Sorry.
Chromosomes are these structures that you can see here. We can see them under the microscope. They're all in the nucleus of a cell.
Okay.
And we get 23 of our chromosomes from mom and 23 from dad. And each chromosome contains genes all along them. So the chromosomes are almost like, drawers, the body's way of storing genes.
Okay. So those genes that we were talking about that are 12,000 letters long, those are stored in the chromosomes.
Yes, that's correct.
Got it.
So see here, chromosome 9? Well, it turns out that the GALT gene is on chromosome 9.
Is it on chromosome 9 for everybody?
Yes.
Okay.
Yeah,
okay.
Everybody.
Everybody has it on 9, which we see chrome 9.
Right. And then next to that it says GRCH37. Okay, so you heard about sequencing the human genome, right? That was done a long time ago.
Right.
But in fact, there are edits to the human genome over time. And so this just tells you what edited version they're using.
It's like what version of the book?
Yes, that's exactly right.
Okay, got it.
And
so they're using the version of the book, GRCH37.
Yeah, and then the G I guess it's the coordinates on the chromosome, right?
I think so.
Yeah.
So 345483550T more, or just that probably is the coordinate of the chromosome.
Yeah, and you can see it's very unwieldy, which is why we prefer the C.584T to C. Like, I actually don't really see this third line on that many reports, quite frankly.
Huh. Okay. So that's all. I mean, it's starting to make sense now. These are ways to identify exactly which type of variant or what, minute, because it sounds like galactosemia can present itself in a number of different variants depending on which ones you have, and this is just detailing like which one this patient has.
And it's standardized to make sure that if you get that report from one laboratory and you take it to a different laboratory or physician, that they'll be able to understand it. Because before it was standardized, people would start sometimes for some genes counting in different locations or stopping in different locations. And so it's got its quirks, but it is a standardized way for us to communicate these results between
Yeah, I guess,
parties.
I jest and make fun of the consistency, but at least we have something.
Yes.
At least there is some consistency.
It's consistent. It doesn't necessarily make sense all the time.
Right. Okay. Good to know. All right. So let me get back to my outline real quick. That was so helpful. So thank you for walking us through all of those nuances. We've mentioned that there's different, there's L195P, there's Q188R. Do you know, either of you, and maybe you do, maybe you don't, I don't know if we even know this as a society, know how many variants can result in galactosemia?
I have no clue.
No.
Okay.
Quite a few. There's some common ones. You've hit several of the common ones, but I think there's a lot more, and we still find new ones too. So
that's what I was going to say. Is it feasible to say that there's ones we don't know about out there?
Absolutely.
Yes.
And would there be some that, I guess the way it might work, and correct me if I'm wrong, is like one variant paired with a variant. There might be a variant like L195P that if it's not paired with this specific L349R, it might be paired with something else and not create galactosemia?
Yes. Yeah, you could have it either paired with something that has less impact on the protein, or you could also have it where it's paired with something that's benign and they're just essentially a carrier.
Got it.
Yeah, so for example, for this individual, right?
Uh-huh.
One of these variants came from mom.
Uh-huh.
And the other variant came from dad.
Uh-huh.
Right? But they're carriers. They're not affected with galactosemia.
Right.
Right?
So the mom or dad that has the L195P has that in them, but they just have a second gene that's not paired with it to make
Right.
galactosemia.
That's a normal functioning gene. So this is what I brought this figure up because I think this is important. So remember, galactosemia is a recessive disorder.
What does that mean?
And that means both copies of the GALT gene have to not be working to have galactosemia. So in this example, you'll see the big G stands for a normal GALT gene, and the little g stands for like a Q188R.
Mm-hmm.
So the circle is mom.
Mm-hmm.
So mom is a carrier, and dad is a carrier.
Mm-hmm.
So when they have children, you only pass on one of each pair of your genes. So there are some cases where mom will pass on the galactosemia gene and dad will pass on the galactosemia gene. So that child will have galactosemia.
And this may be getting too much into the weeds, but so, something that I know a lot of parents who are carriers think about after their first child has galactosemia is IVF. And so are we able then, or is our technology able to basically decide which of those genes it's pulling into the egg or the sperm?
I don't think you can decide which one gets pulled. You can test to see which ones are
Right.
there, though.
Oh, okay, got it.
So that's the point of doing the DNA sequencing, right? Because you can't do IVF unless you know what you're looking for.
Right. Okay. So you know what you're looking for. You know what variant you're looking for. You test the ones, and then you figure out which ones have the big G versus the small g. And really, you only have to find it just as long as it doesn't have both small G's.
That's right. So it's for IVF, it would be fine to use carrier embryos.
Oh, wow. Okay. This is so helpful. So helpful. Thank you. And is there, so we call it a recessive disease or disorder. What's the opposite of that? Is there an opposite? Is there like a progressive or I don't know?
It's not really the opposite, but the other form is dominant.
Dominant.
So that you'd only need one copy. And disorders are generally either recessive or dominant. So there's not dominant forms of galactosemia. It's on a disorder by disorder basis.
Got it. Dominant would be like if you had a big G and a small g, you still get it.
Yes.
Yes, that's right. That's exactly correct.
Okay. Feeling smart in this episode.
I was going to say, we're going to hire you soon.
Yeah, we got you.
This you guys are super helpful. All right. So, couple questions here that I have on my outline for the role of the lab. And I think now we're talking less about gene variants and more just in terms of testing as a whole. Tricia, this might be some questions for you as you work in a lab daily. First question would be, is there a national standard for what does get tested, or do some specialists order some tests while others order different variations of those tests?
So in the United States, there are what we call ACT sheets for newborn screening follow-up, which they're not requirements, but they're recommendations for how to follow up an abnormal newborn screen. And so the American College of Medical Genetics publishes those for disorders commonly included in newborn screening. And so there's a classic galactosemia one, which recommends, red blood cell GALT enzyme analysis, GAL1P, and sequence analysis of GALT as kind of, if you think this is your abnormal newborn screening result, these are the tests you do. And then it has branching logic that if they're abnormal, you see a geneticist, you have galactosemia, or what happens if they're abnormal. And so those aren't necessarily required, but I think they're not necessarily, I would say strict, but pretty commonly followed. I think that's what most people do for an abnormal newborn screen that's suggestive of galactosemia. So it's kind of best practices that are recommended, but not required.
Got it. And, you're at the Mayo Clinic. The Mayo Clinic is the lab that my son's blood is tested at. Are there other main clinics? Are there, how many labs are we sending that can do what you guys do?
There's probably I'm not sure how many there would be now. There's definitely more than just our lab that does it. There's multiple big labs that also do galactosemia enzyme testing and Gal1P levels. So there's and a lot of where your testing will end up with is to a certain degree kind of out of your hands, because it'll depend on your hospital and your insurance and all those other, fun factors that everybody has to deal with. But
Yeah.
for all labs in the US, there's accreditation standards. So everybody's inspected. Everybody has standards that they have to use when they develop tests and report them out. So all of those are at a federal level.
I know we don't know the exact number, but or like, we're thinking thousands of labs?
Oh, no.
Of labs?
I would have said like maybe between 10 and 20 max.
I would agree. I mean, I probably could only name 5 or 6. I mean,
Okay.
Mayo is one of the big ones that.
Got it. Okay, got it. And then, when we talk about testing, and Michael, maybe this is more of a question for you. Can parents request more detailed testing? Like, can parents say like, hey, I want to make sure we test for, we're only testing for Gal1P. Can we start testing for Galactitol? Like, is that a patient thing that is okay?
I mean, I think if a parent was interested in that, I wouldn't have any major issues, except I would say, I'm not sure this is really going to help us.
Right. Okay.
But we could certainly, if it's just getting urine, it's
right.
minimally invasive, right?
Right.
So it's not a big deal.
Interesting.
So I wouldn't have a problem doing that, but I think I would discuss with, I'm not really sure we need both.
Right. Okay. Interesting. I guess, and that's, like you said, it kind of comes down to your genetic doctor and who your team is, what they think is important. And like you said, everybody kind of has a different, I know for us, our doctor at UCLA tests, at least so far, has tested for both Galactitol and Gal1P, and he tested at the very beginning the genes. We know the gene I actually understand the gene variations now, so that's great. So I think a part of it is just having the information feels comforting, even if you can't do something with it. Like, having that information feels pretty good. Okay, so we've done a lot of learning. I have some test results from 3 patients, and what I would love to do, I have the permission from the parties. We already looked at one of these examples in the slides, but I have the permission for the parties to share these on the podcast to help, in our efforts of edification. I also have redacted any sensitive or identifying information like names and dates, so we should be all good there. And what I would love to do is take a look at these with the both of you and have you help me understand what I'm looking at, like we've already done so far a little bit. So these are just test results that the patient had at different times. And the first one, it looks like when they were born, had a bilirubin conjugated value of 0.3. Can you help me understand what I'm looking at?
I'm going to let you tackle bilirubin, Michael.
Okay. So bilirubin is a chemical in the body that's formed because of old red blood cells. Okay. So red blood cells only last, 60 to 90 days. And then they get sort of recycled, and the hemoglobin in them gets degraded to bilirubin.
Okay.
And there are 2 kinds of bilirubin. There's conjugated and unconjugated.
Okay.
It's not and that just has to do with chemical groups that are added onto them. And all babies in the newborn period have some degree of elevated bilirubin. Okay. Because babies have a lot of red blood cells when they're born.
Okay.
And so a lot of those need to be sort of recycled. Okay. So not uncommon for babies to get a little yellow, and that's called jaundice, and that's due to high bilirubin.
Okay. And is bilirubin correlated to galactosemia in any way?
Yeah, well, I was getting there. So if if a child has galactosemia and is receiving milk, breast milk, or lactose-containing formula, their bilirubin will go up, and often we see the conjugated bilirubin go up preferentially, and I don't really know why, but I do know the neonatologists are very good about measuring bilirubin levels and looking at conjugated and unconjugated, and if they see the conjugated go up, they will sometimes automatically switch the baby to soy while we're doing testing, because the sickest that a child can be with classic galactosemia is in the newborn period. If it is not recognized and they are receiving milk, they can die. Okay, let's call it what it is. They can die.
Yes, we've said that many times. We hammered that home on this podcast specifically, as it's so important. So a high bilirubin level, would you expect it to decline as the baby gets older and days? And if you see it go up, that's a sign of concern.
So this is not particularly concerning, what you're showing me. I don't know if this is a newborn.
Okay.
The total bilirubin is 5.7, which is elevated.
Okay.
I would really need to know how old this child is.
Okay.
But we don't typically measure bilirubin for the maintenance care of children with galactosemia. Like if a 2-year-old comes in, I'm not going to order a bilirubin.
Okay.
So it's not part of the standard of care.
Okay.
Unless the child is yellow. I mean, that's a different story.
Right. Well, in a lot of, and so we do see that in newborns who have galactosemia who are drinking breast milk. We know that jaundice is a thing that is prevalent, and that would be because of high bilirubin levels. Is that accurate? Got it. Okay. Yes, you're shaking your head. Yes.
And the yellow can't tell you whether it's conjugated or unconjugated. You have to do the lab test.
Okay. Got it. And then, so moving on now to the next report we have, it looks like this is a Mayo Clinic GAL1P test. The age is 4 days, so this patient was 4 days old. Tricia, can you help me understand what I'm looking at now?
Sure. So up at the top, we just kind of have what we call demographics. So you see how you identify your child or yourself, patient name, date of birth. There'll be a whole lot of other numbers on there, like order numbers, client order numbers. That's how we keep track of them in the lab, because we'll barcode everything and then we can scan it to keep track of it instead of trying to match everybody's names all the time. So we have identifiers that we assign. And then also that we make sure, because at every step when we're looking at it, we're going to want, 2 identifiers to make sure that tube is the right and it's followed through this entire process. So we have all sorts of double checks to make sure that we're keeping everything straight. And then at the top, we've got our test code, which is GAL1P, GAL1 galactose 1-phosphate in erythrocytes, which are the red blood cells. So when a tube of blood is collected and sent to us for this testing, we're going to wash it and extract just the red blood cells and measure galactose 1-phosphate only in the red blood cells. That's what most labs use. We are bringing up in dried blood spots, which we think will bring the option for potentially home monitoring or kind of easier collection that you don't necessarily have to go to a hospital for. So we're moving into that, which will bring a few changes, but I think potentially is also helpful for people, gets them out of the big blood draw with needles and venipuncture.
Can we focus on that for just a second?
Absolutely.
I want to make sure I understand what you just said. You're working on home tests for GAL1P?
It would be able to be home collected.
Home collected.
And then
not
mailed. Yeah, not tested, but mailed.
Got it. Would it still be blood?
It would be blood, but you could do it from like a finger stick, kind of similar to how they do the newborn screening test from a heel prick. You can do it from a finger. So like if you donate blood and they do a hemoglobin from your finger stick, it's that similar process.
Okay, great. And so a lot of identifier stuff at the top there. Yes. And then what we see is galactose 1-phosphate RBC 103.0 mg/dl
So, yeah.
high.
Yeah, milligrams per deciliter, that's
Okay.
just the units. So milligrams is kind of the weight, and deciliter is the volume. So how much galactose 1-phosphate by weight is in a set volume of red blood cells.
Got
So
it.
similar
And
to how when you look at anything and there's a concentration or a weight on anything that's labeled, that's what we're basically reporting out here. And then high is just to draw your attention that it is abnormal and flagged. So for a lot of our tests, we have high and low reference ranges. For galactose 1-phosphate, it's only abnormal if it's elevated. So we don't have a low cutoff where we'd say it's abnormal. And then below there, you see the reference ranges. So normal range, that's completely unaffected individuals walking around. Where do they sit for a galactose 1-phosphate in their red blood cells? And that's less than 0.9, so much lower. And then when we have that therapeutic range, that's where we, based on literature reviews and treated patients and kind of our own work, that's where most treated patients most individuals with classic galactosemia on a lactose-restricted diet will fall under 4.9.
okay, and so this patient at 4 days old has 103 milligrams of GAL1P per deciliter of blood, of red blood cells?
Of red blood cells, yes.
Of red blood cells in their system when a normal person at 4 days old would have 0.9?
Yes.
Got it.
So
okay.
and this is typically what we'll see with 4 days is probably pretty close for whether that was initiated because it was a newborn screen or whether they were sick and went to the hospital and they were very quickly calling to see if there was a newborn screen result available.
Got it.
Some states would have it back. Some states might be still finalizing it, but this is typically what we would see in classic galactosemia where they've drawn the testing and started right away.
Got it. And then the additional information, liquid chromatography tandem mass spectrometry?
Yes. So one of the regulatory requirements that we talked about a little bit earlier is you have to explain how you did the test. So that's just the methodology that we use to measure it. So we separate it out from anything that looks similar, and then we measure it against a set quantity so we can accurately tell you how much was in that sample.
Got it. And one question when it comes to it just occurred to me, when you were talking about how you guys test for it, one thing I've experienced, I know a lot of parents that I've talked to experience, is false positives. And because galactosemia is such a rare thing, we were told by our pediatrician that it was probably a false positive on the newborn screening. I know a lot of people are told it's probably a false positive, don't get worried. And now looking back, when all of us parents go like, that's the stupidest thing you can tell me, because it wasn't a false positive and it led to some very dangerous things.
Well,
how do you guys view that?
So I will say that so for many of the disorders that we screened for, we do see a lot of false positives.
Okay.
And with galactosemia, one of the challenges, Tricia could chime in here, is in the summertime, in the hot states.
Yeah. That's specifically what we heard was that it's summertime, it's hot. We see a lot more elevated false positive galactosemia tests because of that. So this is probably a false positive.
So yes, we see that. Now, again, I don't know what your child's newborn screen looked like.
Sure.
So, in a situation like that, the important thing is to look at, the galactose 1-phosphate. I don't, Tricia, do all states measure it on their newborn screen?
No. So most states, for most states when they newborn screen, their first step is the enzyme assay. So they're going to measure GALT on the dried blood spot. And that's what's really sensitive to temperature. So you can get falsely decreased GALT in a lot of cases. Most states follow that up with a galactose measurement, not galactose 1-phosphate, just straight galactose. And that can be helpful because if it's a false positive due to temperature, that's almost never elevated. But every state has a slightly different way of reporting out screens. So there's some that would report out even the low enzyme with normal GALT and with normal galactose, and those can end up with a different algorithm. They might do a repeat screen if the baby's well, and that might be why pediatricians are saying, we see a lot of false positives. Typically, that combination of low GALT plus elevated galactose is galactosemia, but that's often also a nuance that is lost if you don't live in this world and see it all the time. Because
if you don't see it and and our pediatrician was very upfront with us when it's like, I've never seen a galactosemia patient before. And I would say most pediatricians probably haven't because it's so ultra rare, right? So, but you're saying this specific newborn screening, whatever you were saying, that like the heel prick, the blood spot, that is, it can be modified by temperature, or it can be...
Yeah, the first tier test, the enzyme assay, is very sensitive to temperature.
Okay. Do you see a lot of, or do you know if we see a lot of, like, what constitutes as a lot of false positives in galactosemia? Like, are we seeing more than we are seeing actual positives? Like,
yes.
Is it... Okay.
Yes.
So
when they say we get most of these are false positives, they're not wrong. We just would like to discourage that because some of them are true, and that's the tricky thing.
And I guess it's like, I don't know if we have this information, but is it like for every 100 false positives we have an actual positive? Is it every 5 false positives? Like,
well, go ahead, Michael.
I was going to say states have that data. Like, we can look at our newborn screen for galactosemia, and the fact of the matter is the positive predictive value. In other words, if you have a positive newborn screen for something, are you likely to have the disorder? For many disorders, no. But again, the way the newborn screen works, you could screen for something and be at low risk, or you could be at medium risk, or you could be at high risk, right?
Yeah.
And so we try to stratify them.
Yeah, and I guess being on the other side of it, if it's for every 1,000 false positives, there's an actual positive, it makes me feel a lot better being told that it's probably a false positive than if it's for every 5 false positives, there's a real positive. Like, because then I'm like, well, it's not actually that many.
Well, I think to be fair, as long as while they were doing the follow-up studies, your child was on soy.
Well, and that's the key is we and some other parents I've talked to, many other parents I've talked to, where you can stay on breast formula or you can stay on breast milk for now. And when we get the confirmation back, we'll change them then. But it doesn't seem like he's doing too bad now. And we think it's a false positive. And babies normally get jaundice when they come out of the womb. So he's a little bit jaundiced, but it's not anything crazy. He's a little lethargic, but newborns are lethargic. And it's just a really dangerous thing. And what I advocate for on this podcast is all the time is even if you think it's a false positive, like, why not switch them over to soy? You can always go back to breast milk.
Right. And so one of the things that we try to do, and hopefully all of the physicians here are doing this, especially there are families who really want to breastfeed. And what I advocate is, look, just switch to soy for now, pump, store your breast milk. And if it's not galactosemia, you've been pumping, you can start breastfeeding again and, call it a day.
And that, I think, is the in an advocacy point of view, that's what I try to advocate even to any doctors out there that like maybe come across this podcast. Is like, it's so important. And I know it's not happening everywhere like that because it didn't happen to us like that. And we live in one of the most major cities in the world, and one of the most prominent hospitals is the one my son goes to. So if it could happen to us, it could happen anywhere to anybody. Let's keep going with these test results because I know, we don't have a ton of time and we're already at over an hour. I want to get to... So we went over this a little bit. This is a full gene analysis that was done at the Mayo Clinic. And it looks like...
Yeah, that was the one we went over.
I think this is the one that Michael had in his example.
Yeah, great. So this one, if you're watching on YouTube or watching along, this is the actual test result that Michael pulled from, which has the variance where the the code for position 584, the T has become a C.
Yep.
And that in combination with the other gene having a code that at position 1046, where the T has become a G, has created galactosemia.
Yep.
Perfect.
Freaking parents are going to love to actually understand this. And then this page, Tricia, is just, it looks like it's just a recap of what we found.
Yeah, there's kind of a verbal summary of saying, what other testing do you still have to do? Because you still have to, in some cases, confirm that, yes, you found two variants. You often have to confirm that they are one from mom and one from dad, because in some cases you could inherit two from one parent. Less common in galactosemia, but it's kind of one of the things you do still have to confirm if you don't have other supporting evidence. If you have enzyme that's consistent with it, that's very also very strongly supporting that they're inherited from each parent. And then the references that were used, and again, this methodology section that we have as well.
Got it. That makes sense. The genes that were analyzed, the disclaimer, that's just some more stuff to read.
It's a very long report.
Yeah. Variant evaluation and test classification, anything worth noting in there?
No, that's kind of just how the lab is doing and this criteria that they're using. So that'll be on every report in some format or another.
Got it. And then it is signed off on, been released by, and I guess sometimes this name is your name.
Not for molecular, but for a lot of the GAL1Ps and GALTs, yes.
Got it.
That's one of my colleagues.
Got it. We'll tell him thank you very much.
Will do.
Okay, and then let me just scroll to one we haven't gotten to yet, which is the Galtol, the Galactitol. So this was at age 6 months. The same patient was tested for Galactitol levels, and they were found to have 325 mmol per mol/cr.
Yes. So that is, when we measure something in blood, we can do it by volume because blood has a fairly consistent makeup in an individual. When you measure a urine samples are very dependent on hydration and a lot of other factors. So we normalize a urine specimen to the creatinine value, which means it's much easier to compare from sample to sample because obviously, if you chug a whole bottle of water because you have to produce a urine sample, it's going to be more dilute than if you weren't. And so it can be really, it adds a source of variability that we don't like to have when we're providing test results. So every time you see that kind of per mole creatinine, and that's what the CR is, that means that we've standardized that so that we're reporting it out kind of independent of that person's hydration status at the time. And so then the 325, in this case, we've done it as moles, which is back to your high school chemistry. We're going to get you back to chemistry. You talked about moles and molar weights and things like that. It's just kind of a convenient unit when we talk about, actually, I don't know why we always use mole instead of grams. It's just, we have to measure it in one way. And so we clearly didn't standardize this either. So, it's moles of millimoles of galactitol per mole of creatinine.
Got it. And then your reference value to the right is saying that typical 6-month-old at this without galactosemia is under 109.
Yes.
Got it.
And then it says they're abnormal, and then we also have kind of adding in that what we know about galactitol in treated individuals. So this is somewhere between a normal individual who doesn't have galactosemia and an unrestricted diet in galactitol, which is greater than 8,000 millimoles per mole creatinine. So that's kind of the equivalent when we saw that 100 on a GAL1P that was collected at 4 days old. That's when we're looking at that 8,000 range is what we'd see in galactitol.
And if you got something that was an 8,000 range, that would be something that's concerning.
3 or 4 day old, we'd assume that it was a follow-up newborn screen and they were doing that. In a 6-month-old, we'd probably be calling to say, did you guys know about this? Were you expecting it? Sometimes we have cases where they're doing testing because they know, whether the child wasn't necessarily with their parents or whether they were with a family member and their diet wasn't necessarily perfectly dialed in. And they're like, hey, this is going to be elevated. We know they weren't. We just want to get a baseline of where they're at. So sometimes we have that information, but if it's really out there, we're going to be calling somebody to make sure that they know about this. We don't want it to, slide into Epic on a Friday afternoon and nobody take a look at it.
That's right.
Yeah. Okay. And so I've got 2 more patients here, so I want to kind of quickly breeze through these if possible.
Sure.
I've got a second patient here who also happened to go to the Mayo Clinic. And so a lot of this stuff is going to look very similar, but there were a couple of things I wanted to ask about. So one of these, at age 10, this patient was given a GCT test. It says galactosemia reflex blood, and we see galactosemia reflex B 0.0. What are we testing for here? Oh, look, this is you. You did this one.
I did that one.
Funny that.
So the when GCT is just a different way of ordering the test. So we have it so you can order GALT enzyme and you can order molecular separately, or you can order GALT enzyme, and if it is abnormal, we will reflex to molecular testing. So some providers prefer it, some don't. So it's just an opportunity to order one test, and if it's abnormal, you'll get the molecular confirmation, and if it's normal, you won't. So it's just a way to kind of streamline that process of, we're going to do the enzyme so you know if they have galactosemia or not, and then if you need to, we're going to do the molecular testing so they have that additional information.
Okay.
It's kind of just a different option on the menu.
And if you look at the top, there's no, go back. It says galactosemia reflex B, and then underneath that it says GAL1P uridyl transferase.
That's why we say GALT.
That's the fancy term for GALT.
Okay, got it. So what are we looking at here? What is 0.0?
0.0, so that's a measure of the enzyme activity. So what we do in the blood sample is we add a labeled galactose 1-phosphate that we can tell the difference of between natural galactose 1-phosphate. And we're going to let that react and then measure the product.
So you introduce Gal1P into that blood cell and say into what?
Into the blood sample, yes.
The blood sample and see if it can transfer it into what a normal body would. And at what level it activates?
Yes.
And this patient had a 0.0. So in, let's say, someone with Duarte galactosemia, it's not 0.0, is that right?
Yes.
Or a variant of galactosemia, classic galactosemia is 0.0.
Yeah, classic galactosemia, I believe our cutoff is less than 1.
Okay.
And then you see our reference value there of greater than about 24 or 25.
Right.
And you can kind of, galactosemia is one of the few enzymes that's nicely additive. So if you have a classic galactosemia who's at approximately 0, carriers for classic galactosemia will be around 12.
Oh.
And Duarte galactosemia, which is a D and a G, will be around between 5 and 10. And then carriers for just a D, so DN, usually around 15 to 18.
Okay.
And above.
And then non-carriers
greater than
above
25,
24,
yeah.
25.
Yep.
Got it.
So I'm
actually probably like 20. I'm probably like 12.
You're probably in that ballpark, yes.
Interesting. And that does enough in my body to turn the Gal1P into what it needs to turn it into.
Yes.
Huh. Well, I'm just learning so much, guys. I love this.
Glad it's helpful.
And we've got a Galactitol test here, which we already talked about, 436. Galactitol, okay. And then this is a test I hadn't seen before. That we haven't looked at yet. Is this anything different than what we have been looking at?
It's just a different presentation, but that's the Gal1P as well, yes.
This is the results just presented differently.
Correct, yeah. And there's
Oh,
different units. So you can see milligrams per deciliter. And then we got micrograms per gram hemoglobin, which is another way of standardizing for red blood cell assays.
okay. Got it. And last but not least, one of our friends in Europe sent us their test results, and it's obviously, no surprise, it's quite different presentation. And so maybe you can just do your best. I know this isn't a Mayo Clinic test, but we can kind of, knowing what we know, take a look in, because I know we have a lot of people that live in Europe and watch this as well. What do you kind of see here from your knowledge that makes, is transferable to what we know?
I would say that's when you see that GAL1 PUT, that's the enzyme assay. So
Okay.
galactose 1-phosphate uridyl transferase, and 0.2 is low. They don't have any units, but that's low by almost any standard that you've got. And then I suspect this looks like somebody who did not have a positive newborn screen. They look like they came in sick. I would agree with that, Michael.
I can confirm that that's the case.
Okay.
Well, I think is this from the UK? Because I don't think they screen for galactosemia.
I don't think they do.
It is from the UK, and I can confirm they do not screen in the UK. We've had a couple of people on the podcast who both of their children had to go in being sick and not having a newborn screening test. Do you have any idea of what these other things are on there?
Well, I mean, so they do have LFT. Those are called liver function tests.
Mm-hmm.
And I don't know what max ALT 237, but 237 seems high to me.
I would say that maybe like that is their the max they. That
Yeah,
probably
the lab's
which is
upper
high,
limit. Yeah. And then the bilirubin, I don't know what these units are because we don't usually see 385 as a bilirubin, and the conjugated is 36. So I don't know what that means. I'd have to. And then the
and then it looks like some details here were admitted on day 7 of life was 12% loss of birth weight, poor feeding, and jaundice. And this person, so this result would just be what happened with this person.
Yeah, so they say, right, so the LFTs and the INR worsened. So usually, I mean, my understanding is that our British colleagues are, they recognize galactosemia better than the U.S. pediatricians.
Right.
And then so because of abnormal ALT and clinical state, sure. So they made sure the baby wasn't infected, but vomiting was significant, had low blood sugar, feeding stopped, full septic screen, clinically started to improve, was discussed with the liver unit. Because of potential diagnosis of galactosemia before results received from lab for the enzyme, progesterone feeds given and breast milk normal formula avoided, right? So they took them off breast milk.
Yeah.
Yeah.
Okay. And then they put him on progestamil, which I imagine is the formula. And then here we've got some results. And it looks like they did a GAL1P uridyl transferase.
Yes, that's the enzyme.
Okay, that's the enzyme. So they're testing the level?
Yes. And so their patient was 2.6 by their method, which is a little bit different from ours. And then you can see it falls into that bottom one there is their galactosemic range.
Got it. And so that's really just their testing of does this patient have galactosemia?
Yes.
Okay, yeah. So, and this is, I only had one test result from the UK, so it seems like things are presented a little bit differently over there, but we're testing for the same thing.
Yes.
Got it. Man.
The biggest difference would probably be the units will vary a lot between countries.
Got it. Okay, got it. But they will have their own ranges which
Yes.
in their units
Yes.
would fall in that would have somebody have
Yes.
galactosemia. Wow. I mean, I think this might be one of my favorite podcasts that I've done, just because I went in having close to zero knowledge of genetic variations and what we're actually testing for. I know Galactitol is a thing and Gal1P is a thing, but this adds so much clarity to these test results. And I specifically think of when a new parent finds and stumbles upon this podcast, hopefully this can bring a little bit of like, we often say information is power, knowledge is power on this podcast. And just having the knowledge of being like, okay, I at least understand a little bit of what these tests say is very, very powerful. So I want to say thank you to Michael and Tricia for coming on and explaining it to us. Is there anything that we missed, anything that's worth going over?
I think you've covered all the high points from a lab perspective.
Yeah.
Okay. Great. Well, I mean, I didn't cover most of it. You guys covered it, but I feel like I could cover it now.
I mean, I think one of the things, though, that people can have a misunderstanding that, oh, well, as long as my child avoids lactose or, galactose, he or she will be fine. And unfortunately, that's not always the case.
Mm-hmm.
And we don't fully understand that.
Mm-hmm.
And quite frankly, there aren't enough people working on galactosemia, and I'm glad that Judy Friedevich-Kyle has really kept working on it. So I think we need to understand why. I have two brothers I follow. I mean, they have classic galactosemia. They are smart kids.
Mm-hmm.
I mean, they have no language apraxia. They're doing great. I have another patient. She struggles in school.
Mm-hmm.
And, we don't know why.
Yeah, that's one of the things that we've talked a lot about in our previous episodes, is just that you really do see a wide range and a large spectrum of how galactosemia presents itself. And it's honestly one of the reasons why I started this podcast, is because when you become a new parent, you are told a lot of times that your kid is going to have disorders, your kid is going to be delayed, and you go online and you look up videos and you see some pretty severe cases of delays with galactosemia, and you become worried, when the truth is, like you said, it presents itself in everybody differently, and we don't know why, and we can't really tell by the levels at the beginning if somebody's going to be more delayed or less delayed. So really creating that super huge fear in the beginning can be dangerous for a new parent, because it might not be true, and it might be true, and that's the thing, is we don't know, but there are some things we do know and we test for, and that's why it's important to talk about it and go over it. So, that's, I appreciate you guys sharing your expertise with us and your knowledge. Thank you guys so much. I think I can speak for all of our community when we say the work that both of you do is so important, especially to us. And so we really, really appreciate it. And for giving me your Friday afternoon and spending some time with me as well. I appreciate it.
All right.
This has been really, really interesting. I'm happy to help. If this was helpful, I hope somebody found it. Yeah.
Yeah, I'm sure. I know for a fact that families will find this impactful and helpful. So thank you so much, and thanks for listening to this podcast. Michael, Tricia, have a wonderful weekend, and we'll talk to you soon.
Thank you so much.
Bye now.
Bye.